Usually, when we talk about human anatomy, there's this default expectation of solid architecture, right? You tend to think of the body like a house.
Right. You picture the structural beams.
Exactly, which are the bones.
Right.
And you have the plumbing, like the blood vessels. You have the wiring, the nervous system. You can point to them. They're these distinct, tangible things.
Yeah, it's a very mechanical way of viewing ourselves, and it's heavily biased toward what we can, easily see when we open a textbook. We look for the distinct organs and the discrete systems.
But then you look a little closer at the sources we are exploring today for this deep dive, and you realize that holding all of that macroscopic architecture together, like separating the wiring from the plumbing, keeping the whole biological house from just, I don't know, rotting away or collapsing into a disorganized puddle, is this incredibly complex network of invisible boundaries.
Yeah, secret linings that most of us really never even think about.
Right. Okay, let's unpack this, because when most of us hear the word membrane, we probably just think of skin.
Or maybe, like the eardrum.
Yeah, exactly. But these thin sheets of tissue are doing an absurd amount of heavy lifting for you. They are the ultimate border patrols, protecting, secreting, absorbing, and filtering every single thing that moves through you.
They really are. And in standard histology, there are essentially 4 broad categories of these body membranes. You have 3 that are classified as epithelial membranes.
Okay.
Those are the cutaneous membrane, which is your skin, then mucous membranes and serous membranes. And then you have one major outlier, the synovial membrane.
The black sheep of the group.
Pretty much.
Yeah.
And if we connect this to the bigger picture, understanding how these 4 specific membranes operate at a microscopic level is basically the ultimate shortcut to understanding how your entire body maintains homeostasis.
Because everything interacts with them eventually.
Exactly. Every disease process, every infection, and every healing mechanism eventually has to cross one of these borders.
But to even, conceptualize these boundaries, we kind of have to rewind a bit. Because human beings didn't always view the body as a collection of specialized layers.
No, not at all.
To understand what these tissues are doing, we have to look at how we figured out they existed as distinct entities in the first place. And that takes us from our nice, modern, microscopic definitions back to a very morbid, profoundly obsessive winter in 18th century Paris.
It is such a phenomenal origin story. The person most responsible for organizing membranes into a formal anatomical concept is a French anatomist named Marie-François-Xavier Bichat.
Right.
He was working in Paris in the final years of the 1700s, and in 1800, he published this groundbreaking work called Traité des Membranes, or Treatise on Membranes.
And based on the notes here, the methodology he used to get this data was just staggering. We are talking about a guy who, by his own account, dissected over 600 cadavers in a single winter.
A single winter? I mean, he basically lives in the dissection rooms.
That is insane.
It is. And unfortunately, he paid the ultimate price for that dedication. He only lived to be 30 years old. He died from what was likely tuberculosis, which was undoubtedly contracted, or at least heavily worsened, by his constant exposure to all those diseased tissues he was studying.
Wow, 30 years old.
Yeah. But the truly revolutionary aspect of his work is that he did all of this without relying on a microscope.
Wait, really? No microscope at all?
None. This was decades before cell theory was formalized. He used chemical and physical stress testing. He would boil tissues, dry them out, apply acids, and just observe how they decayed.
I want to jump in with an analogy here because it really frames how radical his thinking was. Earlier anatomy, like the stuff going all the way back to Andreas Vesalius in the 1500s, was kind of like trying to understand a car by looking at its distinct parts.
Right, macroanatomy.
Yeah. An anatomist would say, here's the door, here's the engine, here are the wheels. And they viewed the thin linings on those organs as just incidental wrappers. Like, oh, the lung has a wrapper on it, the heart has a wrapper on it.
Like plastic wrap on a sandwich.
Exactly. But Bichat came along and started looking at the materials of the car. He wasn't categorizing by the door and the wheel. He was testing the steel, the plastic, and the rubber.
That is the perfect way to frame it. He proposed that the body wasn't just made of organs. It was made of distinct tissues that recombined to form those organs.
Which is such a modern way of thinking.
Totally. His brilliant deduction came from pure observation. He noticed that the sac around the heart, the sac around the lungs, and the lining of the belly all reacted to chemical irritation and to natural diseases in the exact same way.
So even though they were in totally different anatomical zip codes, doing totally different jobs for totally different organs, the material itself was identical.
Exactly. Which proved to him that tissue type, not the organ itself, is what dictates pathology.
The material matters more than the shape.
Precisely. The membrane dictates how a piece of the body will behave, how it heals, how it inflames, or how it fails. That realization is basically the direct forerunner of modern cell biology and histology.
So, building on Bichat's realization that the material dictates the behavior, let's look at the actual microscopic engineering of these materials.
Let's do it.
The overarching theme in the source material for the core three, the primary epithelial membranes, is that form perfectly matches function.
Always.
The structure of every single membrane is brilliantly over-engineered for whatever environment sits on the other side of it. Let's start with the cutaneous membrane, our skin.
This is a really unique one. It's the only membrane in the body built to be entirely dry.
Because it's on the outside.
Right, because it faces the open air. It is constantly subjected to desiccation, thermal stress, friction, and just a massive barrage of pathogens.
Just a terrible neighborhood to live in, biologically speaking.
Oh, the worst. To handle that, its outer epithelial layer, the epidermis, is constructed as a stratified squamous epithelium. That basically means it has dozens of layers of flat, scale-like cells.
And those cells are essentially committing a highly coordinated suicide as they move to the surface.
Right, it's one way to put it, yeah.
I mean, they fill themselves with keratin, which is this tough, fibrous protein, and they die, creating this tough, waterproof biological armor.
Exactly. It's built like a heavily reinforced shield.
But as soon as we move inside the body to passages that are exposed to the outside world but aren't actually in the open air, that heavy armor would be a huge liability.
Right. You can't absorb anything through a dead shield of keratin.
Exactly. So the engineering completely changes when we get to mucous membranes.
Mucous membranes, or mucosae, line all the internal passageways that eventually connect to the outside world. So we're talking about the respiratory tract, the digestive tract, the urinary, and the reproductive tracts.
The wet passages.
Yeah. Because they're internal, they don't need that heavy armor. Instead, they need to facilitate active transport. They specialize in secretion and absorption.
I want to pause on the nomenclature here for a second, because the sources clarify a misconception that I definitely held. These membranes aren't actually defined by the fact that they produce mucus.
No, they aren't. It's a misnomer that causes a lot of confusion, even for medical students.
Yeah, like parts of the vocal cords, for example, or certain areas of the urinary tract have very little to no mucus secretion at all. But histologists still classify them as mucous membranes.
Purely based on their location.
Right, because they line a tract open to the exterior and because of their underlying connective tissue structure. So it's an anatomical classification, not necessarily a functional absolute.
Though we should acknowledge that the vast majority of them do secrete mucus.
True.
It's a viscous fluid produced by specialized goblet cells. And a phenomenal example of why that fluid is engineered the way it is can be seen in your trachea, your windpipe.
Yeah.
It utilizes a mechanism called the mucociliary escalator.
It's a conveyor belt in your throat.
It literally is. The mucous membrane there is lined with pseudostratified epithelium featuring millions of microscopic hair-like projections called cilia.
Like little brooms.
Exactly. The goblet cells secrete a sticky layer of mucus that traps inhaled dust, pollen, and bacteria. Then the cilia beat in a highly synchronized upward wave, sweeping that contaminated mucus layer up toward your pharynx at a speed of about 1 centimeter per minute.
And then you just swallow it.
Right, you swallow it, and your stomach acid destroys the trapped pathogens. It's an amazing defense system.
But let's look at the underlying mechanism when that system fails, because that's when we truly realize how fragile this whole engineering setup is. The sources highlight cystic fibrosis.
Yeah, cystic fibrosis provides a tragic but really highly illustrative look at membrane physics. It's a genetic disorder affecting a specific protein known as the CFTR channel.
Okay.
And that channel normally transports chloride ions across the epithelial membrane and into the mucus.
And this is where the osmosis comes in, right? Because biology is all about gradients. Usually when chloride ions get pumped out into the mucus, sodium follows it to balance the electrical charge.
Exactly. Salt is basically moving into the mucus.
And because you suddenly have all this salt in the mucus, water gets pulled out of the cells via osmosis to dilute it. And that water is what keeps the mucus thin and slippery.
You nailed it. But in cystic fibrosis, that chloride channel is mutated. The chloride gets trapped inside the cells.
So the salt doesn't move.
Right. And because the ions can't move into the mucus, the water doesn't follow. As a result, the mucus on the surface becomes incredibly dehydrated, thick, and rubbery.
It just becomes a physical roadblock. Like the cilia are still trying to beat it, but they are trapped in this, well, this biological cement.
Cement is exactly what it feels like. That 1 centimeter per minute escalator completely stalls out. The bacteria you breathe in just sit in the lungs and multiply, leading to chronic catastrophic infections.
And it's all because a single ion channel failed to hydrate a microscopic layer of mucus.
It emphasizes how dependent we are on exact fluid dynamics at the cellular level, which actually perfectly transitions us into the third category.
Serous membranes.
Right. If mucous membranes line the tubes open to the outside, serous membranes line the completely sealed internal cavities of the body.
So the thoracic cavity housing the lungs and heart, and the abdominopelvic cavity housing the digestive organs.
Yes. And think about it. These organs are in constant violent motion. The heart is beating 100,000 times a day. The lungs are inflating. The stomach is churning.
If they were just rubbing raw against the body wall, the friction would literally shred them.
Absolutely. That's where the elegant design of the serous membrane comes in. It always comes in a continuous matched pair.
Right. The sources had a great visual for this.
Yeah, imagine pushing your fist into an underinflated balloon. Your fist is the organ. The inner wall of the balloon, wrapping directly around your fist, is the visceral layer.
Okay.
The outer wall of the balloon is the parietal layer, which lines the cavity itself. And the tiny space between them, the inside of the balloon, is filled with a microscopic film of serous fluid.
The analogy in our source notes that really brings this to life is imagining a thin film of water between two panes of glass.
Oh, that's a great one.
Yeah, like if you press two pieces of glass together with water between them, the surface tension makes them almost impossible to pull straight apart. But if you slide them against each other, they glide with almost zero friction.
Yes, that is exactly what your lungs are doing against your rib cage every single time you take a breath.
It's just wild to think about.
The pleural cavity around your lungs contains only a few milliliters of fluid per side. It is a highly regulated microenvironment.
But of course, when that physical environment is disrupted, the clinical realities are pretty severe.
Very severe. Take a ruptured appendix, for instance.
Right. If the appendix bursts, it dumps highly infectious intestinal bacteria directly into that perfectly sterile, perfectly sealed peritoneal cavity.
And the visceral and parietal layers of the serous membrane immediately become inflamed. That's a condition called peritonitis. Clinicians historically detect this using a physical exam technique looking for something called rebound tenderness.
Which honestly always sounds completely counterintuitive to me. Like, you would assume the peak pain happens when the doctor pushes their hand deep into your inflamed abdomen.
You'd think so, right. The initial pressure does hurt, definitely. But the agonizing spike in pain actually occurs when the clinician suddenly releases their hand.
Wait, on the release?
On the release. When the pressure is removed, the abdominal wall springs back, and it violently stretches that inflamed, highly innervated parietal peritoneum. It's the mechanical stretch of the tissue that triggers the massive pain response.
Ah, so it's all physical mechanics. And you also have situations where the fluid regulation itself fails, right? Not because of an infection, but because of systemic pressure changes, like in advanced liver cirrhosis.
Yeah. In liver cirrhosis, the architecture of the liver becomes heavily scarred. This creates massive resistance to blood flow, leading to a condition called portal hypertension.
Okay, so the blood backs up.
Right. The blood backing up creates immense hydrostatic pressure within the capillary beds of the digestive organs.
And that hydrostatic pressure essentially acts like a squeegee.
That's a very vivid way to describe it.
Well, it physically forces the blood plasma right out of the capillaries, across the visceral peritoneum, and into the abdominal cavity, right?
Yes, resulting in ascites. You take a serous cavity that was engineered to hold a barely perceptible film of moisture, and suddenly it is acting as a reservoir holding several liters of fluid.
Literally visibly distending the entire abdomen.
Yes, but the important clinical distinction here is that the fluid buildup isn't the primary disease.
It's just a symptom.
Exactly, it's a mechanical symptom of the upstream pressure failure in the liver.
But medical science hasn't just figured out how to, diagnose these membrane failures. We've actually figured out how to hijack the architecture for treatment.
Yes, we have.
The sources bring up peritoneal dialysis. We're literally using the belly as a biological filter for people whose kidneys have failed.
It's such a brilliant application of membrane physics. The peritoneal membrane has a massive surface area and is heavily vascularized. So in peritoneal dialysis, doctors introduce a specially formulated dialysate fluid into the abdominal cavity through a catheter.
And this fluid is packed with glucose.
Exactly. It has a high concentration of glucose.
So we are basically creating an artificial osmotic gradient.
Precise.
The glucose in the cavity acts like a chemical magnet. It's pulling the urea, toxins, and excess water right out of the bloodstream, across the serous membrane, and into the dialysate fluid.
Which is then just drained out of the body. We are utilizing the body's own wrapper as a semi-permeable filter.
That is just so incredibly clever.
It really is. Hundreds of thousands of patients manage their end-stage renal disease using exactly this mechanical exploitation of their own anatomy.
Okay, so we've established the rules for the core 3 epithelial membranes. They all have an epithelial layer anchored to an underlying connective tissue layer. They manage borders.
Right.
But biological science hates a perfect rule system, which leads us to the 4th category, the outlier, the synovial membrane.
The exception that proves the rule.
Here's where it gets really interesting, because I kind of have to push back on this classification based on the reading.
Oh, really? Let's hear it.
Well, the synovial membrane lines the capsules of our freely movable joints, the knees, elbows, shoulders. And it secretes synovial fluid, which has the consistency of raw egg whites, right?
Yeah, to lubricate the joint and nourish the cartilage.
Right. But it breaks every structural rule we just spent 10 minutes establishing.
It does.
It has absolutely zero epithelial tissue. It is constructed entirely of specialized connective tissue. So if it lacks a defining structural component, why is it grouped with the other 3?
It's a fair question.
And while we're poking holes in anatomical classifications, what about the meninges? You know, the dura, arachnoid, and pia mater wrapping the brain and spinal cord.
Right, the brain's wrappers.
The word meninges literally translates from Greek as membranes. They separate compartments, yet they are excluded from this specific Big 4 classification.
You're touching on a really fundamental tension in histology here. It's the debate between structural definitions and functional definitions.
Okay, explain that.
Well, if we're strict structuralists, a true membrane must possess an epithelium, period. But functionalists argue that biology is defined by what it does, not just what it's made of.
Form follows function.
Exactly. What's fascinating here is that over time, the functional definition won the textbook wars for the synovial membrane, but it didn't for the meninges.
Why did synovial make the cut?
Synovial membranes are included because they behave like serous membranes. They encapsulate closed cavity, they regulate a very precise fluid microenvironment, and they mediate transport.
So it's basically an honorary epithelial membrane based purely on its job description.
Yes, exactly. And understanding that functional vulnerability is critical when we look at autoimmune pathologies, like rheumatoid arthritis.
Because the immune system gets confused.
Right. In rheumatoid arthritis, the immune system fundamentally misidentifies the synovial membrane as a threat. T cells and B cells infiltrate this connective tissue boundary, triggering a massive inflammatory cascade.
The synovitis.
Right. But it doesn't just get inflamed. The synovial membrane aggressively thickens and proliferates. It transforms from this delicate lining that's just a few cells thick into a massive invasive tissue mass called a pannus.
Wow. A pannus.
Yeah. And this pannus begins secreting enzymes that actively digest the adjacent articular cartilage and the underlying bone.
Oh, wow. So the membrane itself weaponizes and destroys the very joint it was designed to protect.
Exactly. If rheumatologists didn't understand the specific cellular behavior of that membrane, the entire mechanism of the disease would be a total mystery.
It always comes back to border control.
Always.
But because these functional definitions are so microscopic and the physics are so abstract, the historical record is just littered with spectacular misunderstandings of how these tissues actually work.
Oh, absolutely.
Which brings us to the medical myths highlighted in our sources. Let's talk about the tennis court myth. Because I guarantee almost everyone listening to this was taught this in high school biology.
Oh, definitely. It was everywhere.
It was presented as an absolute, undeniable textbook fact that if you took the mucous membrane of the human small intestine and you flattened out all the microscopic folds and the villi, the surface area would equal the size of a tennis court, roughly 200 to 300 square meters.
It was an incredibly sticky piece of trivia. It survived in physiology textbooks for half a century.
But the sources point out that in 2014, two gastroenterologists, Herbert Haylander and Lars Fendrix, decided to stop repeating the fun fact and actually check the math.
Which is what scientists should do.
Exactly. They used modern microscopic cross-sections to recalculate the geometry, and they found it's nowhere near a tennis court.
Not even close.
It's about 30 square meters, roughly the size of a small studio apartment.
The earlier anatomists had basically assumed a perfect, dense fractal geometry. They calculated the macroscopic cylinder, multiplied it by the circular folds, multiplied that by the villi, and multiplied that by the microvilli, assuming maximal density everywhere.
Just assuming it was perfectly packed.
Yeah. Helander and Fendrix proved the mucosal surface is actually much more irregular and far less dense than the models assumed.
It is an order of magnitude smaller than the folklore version. It's just wild to marvel at how a quotable, mathematically flawed number was blindly passed down by credentialed scientists for generations before anyone bothered to verify the variables.
It serves as a powerful reminder of the inertia of scientific consensus, that's for sure. And sometimes believing the wrong thing about a membrane surface has much deadlier consequences than just a bad trivia fact.
Right, moving from bad math to bad medicine.
Exactly. Before the acceptance of germ theory, deadly inflammations of these membranes, like the peritonitis we discussed earlier, or puerperal fever in postpartum women, were heavily blamed on miasma.
The concept of toxic infected air.
Right. The establishment believed the air itself was causing the tissues to rot. They were completely missing the mechanical reality that microscopic bacteria were being physically transmitted across these wet, highly vulnerable surfaces.
And the ultimate tragedy of this paradigm is embodied by a Hungarian physician named Ignaz Semmelweis, right?
Yes. In 1847, working at the Vienna General Hospital, Semmelweis noticed a stark discrepancy. The maternity ward, staffed by male doctors and medical students, had a catastrophic mortality rate from puerperal fever.
But the other ward didn't.
Exactly. The ward staffed by female midwives had a much lower rate. He deduced the crucial variable. The doctors were routinely going straight from performing autopsies in the morgue to delivering babies.
Oh my God, they were carrying the biological material of corpses directly onto the highly vascular, easily traumatized mucous and serous membranes of these women during childbirth.
It's horrific. So he instituted a mandatory intervention. Doctors had to scrub their hands with a chlorinated lime solution before touching any patient.
Even though he didn't even know what bacteria were yet.
Right. He didn't know the microscopic exacts, but he knew the chemical wash disrupted the transmission. And maternal deaths absolutely plummeted to less than 1%.
That is incredible.
He practically eliminated the spread of infection across those membranes in his ward.
And for figuring out the physical mechanics of transmission and saving all those lives, he was run out of the hospital.
He was viciously ridiculed.
Yeah.
The medical establishment found it highly offensive to suggest that a gentleman physician's hands could transmit disease.
Because their hands were supposedly inherently clean.
Exactly. His data challenged the miasma theory too aggressively. He was ostracized, lost his position, suffered a nervous breakdown, and ultimately died in a psychiatric asylum decades before Louis Pasteur and Robert Koch formalized germ theory to vindicate him.
Wow.
This raises an important question, though, regarding how we approach medical facts.
Yeah, it shows a massive cognitive dissonance in our own understanding of the body. Semmelweis observed the physical reality of membrane transmission. He had the statistical data to prove it, but the prevailing scientific paradigm simply could not accommodate a truth that contradicted their established worldview.
The facts were entirely subordinate to the established narrative.
It really makes you wonder what obvious mechanical truths we are currently ignoring.
Absolutely.
But as we wrap this up, let's crystallize why knowing the architecture of these invisible borders matters to you, the listener, right now. Understanding these four categories, cutaneous, mucous, serous, and synovial, is basically the ultimate diagnostic cheat code for your own body.
It really is.
Like if you get a sudden sharp stitch in your chest that spikes with agony every time you take a breath, that friction implies pleurisy and inflammation of the serous membrane.
Exactly.
Your relentlessly runny nose. That's your nasal mucosa ramping up fluid secretion to physically flush out a viral load. A swollen, hot, immobile knee. You're likely looking at synovitis.
Identifying which specific boundary is failing and understanding its distinct microscopic properties is the fastest way to isolate the pathology.
They aren't just wrappers keeping your organs from touching.
No, they are highly active, dynamically regulated biological shields that dictate what stays out, what gets in, and how the entire system communicates.
They are the quiet architecture behind the gross anatomy, keeping the whole biological house from collapsing. And exploring the history of how we discovered them leaves me with this final thought.
Yeah.
If medical science blindly believed that the surface area of our intestines was the size of a tennis court until the year 2014, simply because no one checked the math, and if the establishment literally drove the man who figured out how membrane infections spread into an asylum to protect their egos, what heavily quoted, deeply entrenched textbook facts about the physical structure of our own bodies are we still getting completely wrong today?
It's a great point.
What is the next tennis court myth waiting for a 21st century calculator to finally debunk it?
It guarantees that our maps of the human body are still just graphs.
Hmm.
There's always more to learn.
Always more to learn. Thanks for joining us on this deep dive into the source material. We'll catch you on the next one.